Dear students, it's lecture about nervous tissue. In nervous tissue present two main types of cells. It's neurons and neuroglial cells. Neurons is a main type of cells which provide and form impulses and provide its impulses to main organs. Neuroglial cells, it's a special types of cells which provide support, protection, and regulation of nerve impulses. Neurons. Neurons is a main type of cells in nervous tissue. Neurons are responsible for the receptive, integrative, and motor function and regulate main function of nerve tissue. It provides nerve impulses and generated its impulse. Between neurons form special cell junction with name synapse. Neurons are composed of cell body, dendrites, and just a single axon. Cell body is an area which surround of nucleus and include cytoplasm, some main organelles, more rough endoplasmic reticulum, Golgi apparate, mitochondria, large nucleus inside in cell body, often with a single nucleolus, and to concern processes. Dendrites, it's a process which more branch and similar to tree. And a second type of process has name axon. Axon always one single process, and it may be very long without branch and form some perpendicular branch which has name collateral. Also, cell body has similar name perikaryon or soma. Impulses in neurons always has special direction. It's physiological direction of neuron. To start from terminal part of dendrite, then impulse go to soma of neuron and or perikaryon, and the next continua form special impulse in axon. In axon present special axonal hillock, which will be increase impulse and save impulse. Impulse continua providing to terminal part of axon and release its impulse to special target organs. It may be muscles, glands, or another neuron. In perikaryon present large nucleus, light staining, and its large nucleus is very active. It's a show light staining of chromatin, euchromatin present more, because nucleus is a very active cell to concern produce proteins. Proteins includes in mediators or transmitters, which will be released and form and continue impulse to another neuron or to muscles, to glands, and all. In cytoplasm of perikaryon or soma of neuron present height level endoplasmic reticulum. It's a rough endoplasmic reticulum, more polyribosomes which activate process of protein synthesis, and in this case in cytoplasm present basophilic substances which form from rough endoplasmic reticulum with polyribosomes, and its basophilic substances has special name Nissl bodies or Nissl substance or tigroid. It's substances visible in light microscope after special staining toluidine blue or Nissil staining. We can see and find some granules, blue or purple color in central part of soma, and investigate its components and take some information about neuron. If it's granules visible, good position, it's a normal situation. But in abnormal situation, maybe a decreased amount of granules and will be in pathological stages, in actives of neurons, or a positive formed very dark staining granules and not visible some elements. It can be after pathological destruction of a neuron. It's a first step of apoptosis of neurons. It's a good scheme. Show to you location of main organelles in cell body of neuron. Visible a large nucleus with single nucleolus, and Golgi apparate and granules of rough endoplasmic reticulum with more elements with polyribosomes which formed Nissil substances or Nissil bodies. Also present more mitochondria because a process formation of protein is energy dependent. Here on a slate visible, and you can find granules in cytoplasm and nucleus in central part with nucleolus. It's after staining of chrysal violet. Neurons is normal now on its slate because visible good nucleus, single nucleolus, and granules of Nissil bodies. In neurons present specific components in processes which has name neurofibrils, and neurofibrils include neurofilaments and neural tubules. Together, neurofilaments and neurotubules formed neurofibrils. Neurofibrils are very important to provide transports and two types of directions of its transport. It will be similar to physiological direction of impulses. I repeat and to you, and it's from terminal part of dendrite, then goes to cell body of neuron, and then continue to axon. It's physiological direction. And we have opposite direction from terminal part of axon goes to cell body and then continue to dendrites. It's a process has name retrograde transport. Skeletal components, neurotubules and neurofilaments, not more large, neurotubules near 24 nanometer in diameter, neurofilaments 10 nanometer in diameter, and together form neurofibrils. Neurofibrils can take silver, salts of silver, and in result stay more visible after staining of silver impregnation. And silver impregnation show position and location of neurofibrils in a cell body and in processes. In cell body of neurons, neurofibrils form network or position in different directions and form skeletal of cell body. And in processes, direction will be in one arrangement. It will be parallel layers and helps to provide substances, transport substances from cell body to terminal part of axon or opposite from terminal part of axon to cell body, retrograde transport. Axonal transport very important because provide neurotransmitters or mediators from cell body to terminal part of axon. Neurotransmitter very important to provide impulses to muscles, to glands, and neurotransmitter to divide on some subparts. It may be acetylcholine or adrenaline. It's a activate of process some, or in other mediators, dopamine, serotonin. It's mediators which will be decrease some release processes in target organs, and neurotransmitters to divide on some parts, subtypes, catecholamine, and catecholamines include adrenaline and noradrenaline and acetylcholine. And to repeat once again, anterograde transport direction similar to physiological direction from cell body to axon terminal, and retrograde transport opposite direction. Axonal transport very important and more fast. Rapid transport, it's a near 400 millimeter per day. It takes place in anterograde transport of organelles or some elements in cytoplasm. And retrograde transport, it's a more slowly transport and it's near 0.2 millimeter per day. Neurons to subdivide some groups, neurons which form classification of neurons. Classification of neurons based by amount of processes subdivide all neurons in human organism on 4 main subtypes. Its unipolar neurons contain just one process. Unipolar neurons can be in embryonic period and form neuroblast cells. Neuroblast cells can divide, goes to mitotic divisions, stay before the liver, and form more new neurons. Pseudounipolar neurons characterized by a single process which lie on very short path, and then subdivide its process on 2 different processes which provide function of dendrite and second provide function of axon. In some books, visible information about 2 processes which lie near one by one on short path and then to subdivide. Here we have special name pseudounipolar neurons. Pseudounipolar neurons lie in spinal nodes near the spinal cord, and pseudounipolar neurons formed specific afferent groups of neurons. It's a fast neuron in a reflex arc, or another name sensory neuron. Third type of neurons has name bipolar neurons. In bipolar neurons present 2 processes, a single dendrite and single axon. Bipolar neurons lie in retina of eye and in specific olfactory area in nasal cavity. It's sensory neurons. And maybe interneurons in retina too. Multipolar neurons predominate in a central nervous system composed of more processes, more dendrites, and single axon. Students, it's a good scheme show to you different types of neurons to concern structure, to morphological classification. Here are visible 3 types of neurons. It's bipolar, one single dendrite and single axon. Dendrite continua and form more branch, like tree. Next, second on picture, it's multipolar neuron, more dendrites present and single axon, final part of axon terminal, and pseudounipolar neuron, which include a single short part of process which subdivide on 2 branch elements. It's dendrite and axon. Dendrite visible on its scheme, more branch continua position of branches, and axon goes and form axon terminal. Second classification of neurons based of their function. To subdivide on three main groups, it's sensory or afferent neurons, which receive impulse and provide impulse to central nerve system. In sensory neurons, in terminal part of dendrite, present specific structures which have name receptors. Second type has name motor or efferent neuron. Motor or efferent neurons provide impulses from central nerve system to specific types of organs, its muscles, glands, or in other neurons. And muscles, if it's a neuron, provide impulse to muscles, has names motor neurons. Have name motor neurons. Interneurons, third type of neurons located between sensory or afferent neurons and motor or efferent neurons. Interneurons located in central nerve system or may be located in ganglions of nerve system and provide impulses from sensory to motor neurons or efferent neurons. Maybe interneurons subdivide to concern amount of processes to bipolar in retina, for example, or if it's more processes, multipolar neurons. Students, here on the slide, very good scheme. Show structure of neuron. Please, it's scheme present in your textbook. You can recognize position of dendrites, axon, cell body, location of nuclear body, and axon hillock location. Be attentive in axon hillock, not visible nuclear bodies. Nuclear bodies not present in axon hillock. Axon hillock increase impulse. It's a very important part of neuron. Impulse then goes to axon, long axon, and final represent provide neurotransmitters or mediators to muscles or in other organs. And terminal part of axon has name motor end plates if its terminal part touch to muscle or regulate muscle contraction. Second type of cells in neural tissue has name neuroglial cells. And neuroglial cells provide supporting and protection and regulation of impulses in neurons. Neuroglial cells subdivide on 5 types: astrocytes, oligodendrocytes, SCHWANN cells, ependymal cells, or microglia cells. But to concern classification by size, neuroglial cells subdivide on 2 large groups: macroglial cells, which include astrocytes, oligodendrocytes, SCHWANN cells, and ependymal cells, and microglial cells, second group. Astrocytes includes in group macroglial cells because very large, visible good on light microscope. And astrocyte has name to concern position of processes, star-like cell and Astor star. Astrocytes provide and form very important structure. It's blood-brain barrier. Blood-brain barrier includes in a process of protection of nerve tissue and protection from myelin sheath proteins, which can provide autoimmune reaction. And blood-brain barrier very important to protection from different viruses, bacteria, and regulate providing of nutrition material. Astrocytes, in its case, form special fits, vascular fits, and fits which will be cover of nerve processes of neurons, and astrocytes to subdivide on two main groups. I will represent on next slide. It's a photo from histological specimen after staining of silver salts, silver impregnation, visible astrocytes with more processes which form specific vascular fit near with blood vessel. In astrocyte present neurotubules and neurofilaments, and astrocytes can be visible on light microscope after staining of silver salts. Types of astrocytes. Astrocytes to subdivide on two main types is protoplasmic astrocytes, which lie in gray matter and form a blood-brain barrier between elements of gray matter, between neurons and blood vessels. And fibrous astrocytes, which are located in white matter. Fibrous astrocytes have more long processes, not more branch, opposite of protoplasmic astrocytes. Protoplasmic astrocytes characterized by short processes, very branch, and formed more vascular fits. Next type of neuroglial cells has name oligodendrocyte. Oligodendrocytes, it's a specific neuroglial cells located in gray matter and in white matter in central nerve system. Oligodendrocytes characterized by some processes, not more amount. It's near 3 to 5 to 6 processes in one cell. Each process forms myelin sheath, and myelin sheaths surround of nerve processes, dendrites and axons of neurons in central nerve system. Students, its good scheme show a process formation of myelin sheath and location of processes in oligodendrocyte. On its picture, in oligodendrocyte present 3 processes. Each process formed myelin sheath in central nerve system. Next type of neuroglial cells is SCHWANN cells. SCHWANN cells located in peripheral nerve system and provide similar function with oligodendrocyte, its formation of myelin sheaths, but differences between SCHWANN cells and oligodendrocytes very nice visible. SCHWANN cell without processes, each SCHWANN cell formed just one myelin sheath. Opposite of oligodendrocytes. Oligodendrocytes have some processes, and each process will be formed myelin sheath. In this case, oligodendrocytes formed some myelin sheaths. It's a similar to amount of processes in oligodendrocyte. But SCHWANN cell without processes, and each SCHWANN cell will be form a single myelin sheath. SCHWANN cells include a single nucleus and formed more surrounds rapids of mesaxon. Mesaxon is duplicate of plasma membrane, and its duplicates will be surround of axon or some processes of neurons. It forms some sublayers, concentrical sublayers. In result, form a SCHWANN cell and myelin sheath. Its good scheme show a process formation of myelin sheath in peripheral nerve system. Visible SCHWANN cells, blue color on general picture of neuron. It's axon of neuron which will be covered by SCHWANN cells, and each SCHWANN cell formed myelin sheath, single myelin sheath. Myelin sheath includes some sublayers of mesaxone, its duplicate of plasma membrane of Schwann cell, which surround more layers and form covering layers. On picture also you can see start process. It's will be surround axon by Schwann cell, and then will be a circular movement of Schwann cell and surround and form more layers of mesaxone. In result, in finish, visible axon in central part, yellow color, and more layers of mesaxones of Schwann cells. And then peripheral part of myelin sheath, visible nucleus of Schwann cell and cytoplasm. Ependymal cells derive from neuroepithelium of neural tube and lie in cavities of central nerve system, cover of cavities of central nerve system. Its spinal cord, in spinal cord present central canal, which cover of ependymal cells, and also cover of cavities in brain, and provide movement of cerebrospinal fluid, or another name cerebrospinal fluid liquor, and form its liquor helps to form formation in choroid plexus. Microglia cells, it's a very small cell, appositive of previous cells in neuroglia, and it's a neurological cells which provide phagocytosis like to macrophages. And cells derived from mononuclear phagocytic cells, it's a population of cells present in red bone marrow, view was study. It's monocytes. Monocytes, next step formation of macrophages, and microglia cells with processes, but in period of inflammation, inactivation, processes stay more short, and groups of microglia cells form specific areas of inflammation. Cells characterized like to similar to macrophages, characterized in activation of immune response, immune answer in central nerve system. It's scheme of types of neuroglial cells present in your textbook. Here you can see types of astrocytes, protoplasmic astrocyte in gray matter in central nerve system, fibrous astrocyte in white matter in central nerve system, oligodendrocytes with 5, 3, 4 processes, and microglia cells in non-active period. On previous slides, I told and showed to you specific neuroglial cells which provide formation of myelin sheaths in central and peripheral nerve system. Myelin sheath is formed in central nerve system by oligodendrocytes and Schwann cells in peripheral nerve system. Consists of several spiral layers of plasma membrane, and which surround of neuron process. It may be axon, maybe dendrite also. And continue its position of some parts of neuroglial cells, oligodendrocytes and Schwann cell, but between some Schwann cells and processes of oligodendrocytes formed short space without myelin sheath. Its space has name node of Ranvier. In nodes of Ranvier formed nerve impulses and provide nerve impulse in myelinated nerve fiber by jumping process. It's a jumping from one node of Ranvier to next node of Ranvier. In its case, process providing of impulses will be very quickly, and specific elements of myelin very nice visible after staining of osmium tetroxide. It's good scheme show to you location and position of node of Ranvier, also location of myelin sheath in nerve process, nerve fiber. And if present myelin sheath of nerve fiber has name myelinated nerve fiber. Or if we have not in myelin fiber, myelin sheath has name unmyelinated nerve fiber. In unmyelinated nerve fibers, providing process of nerve impulses more slowly, appositive of myelinated nerve fiber, and here a very nice visible location of Schmidt-Latterman clefts. Schmidt-Latterman clefts, it's an area in myelin sheath which always light staining after tetroxide osmium, and in its area present cytoplasm of SCHWANN cell or oligodendrocyte to concern function. To concern function, cleft of Schmidt-Latterman provides some maybe nutrition material to axon. Students, it's electron micrograph of myelin sheath. In central part visible axon or nerve process which surround of myelin, and SCHWANN cell cytoplasm visible too. Nerve endings and synapse. Nerve endings, it's a terminal part of axon or terminal part of dendrite, and to concern dendrite, if terminal part of dendrite will be formed maybe receptors or synapse. To concern terminal part of axon may be formed a motor ending plate or synapse. And construction of synapse. Synapse formed by two cells. It will be first cell of neuron, first neuron, and second neuron. It's a junction between two neurons. In synapse, recognize three main parts. It's presynaptic terminal, which includes synaptic vesicles with neurotransmitter or mediator. Next, present synaptic cleft. It's a space between presynaptic terminal and postsynaptic membrane. Postsynaptic membrane, it will be second neuron, which includes receptors to neurotransmitter and takes information from previous neuron. In other words, we have three main subparts of synapse: presynaptic part, synaptic cleft, and postsynaptic part. Presynaptic part includes mediators on neurotransmitter, and postsynaptic part includes receptors to its mediator. On its picture, visible composition of synapse, and you can see and recognize subparts. It's presynaptic terminal, postsynaptic region, and synaptic cleft. Red points on its picture, it's neurotransmitter elements. Neurotransmitter can be proteins or some elements, organic substances which are produced inside a neuron. Histogenesis of nerve tissue joined with histogenesis of nerve system in general, because nerve tissue formed of nerve system, and very important, nerve tissue not classified. In nerve tissue we have not subtypes. In nerve tissue we have subtypes of cells, neurons and neuroglial cells. Development of neural tissue from neuroepithelial, it's a dorsal part of ectoderm. The first form neural plate. The next step is invagination of neural plate and form specific neural groove. Neural tube formed after joining of parts neural plate and form cylindrical structure with hollow inside. And from neural tube in future will be formed central nerve system, brain, and spinal cord. Some cells in period formation of neural tube stay between dorsal neural plate and neural tube and then migrate and form specific neural crest cells. Neural crest cells migrate and form elements of peripheral nerve system, form elements of endocrine system, and also includes in process formation of neural ganglions. Neural crest cells migrate and can form a spinal sensory ganglia, form specific sensory neurons, which includes in peripheral nerve system, autonomic ganglia, and melanocytes in skin and in other parts of organism. Regeneration of proximal axonal segment. It's a specific process, regeneration after destruction and damage. And in its slide, you can see process of regeneration. And very important in its period, SCHWANN cells, because SCHWANN cells form special columns, very important to provide regeneration and provide way of regeneration. And a very important formation of elements in peripheral nerve system also. Thank you for attention, dear students. To next lecture.